Biology Part 80, non-specific immune responses. When any microbe penetrates the body's first line of defense, it immediately triggers a series of changes in inflammation at the point of the entry. Once the most important changes is to release an histamine from the connective tissue, mast cells and other tissue cells. Histamine causes the local blood vessels walls, which allows proteins to leak out into the tissue fluid. The damaged tissues swell, and as the inflammation develops, the effects of the histamine are enhanced by other chemical mediators, including kinins and complacements, and they are proteins formed on the blood plasma, and it continues. In fact, these chemicals attract the neutrophils by a process called the chemotaxis, and they in fact get along with it, and they complete the process, and they use the abscesses and granula to combat the disease. Then there are specific immune responses. If we talk about the specific immune responses, they depend upon the production of antibodies. In lymphocyte cells, Y-shaped proteins with the structure illustrated. The molecular consists of the four chains comprising of two heavy chains and two light chains linked up with the disulfide intriges. Part of this, it provides the terminal portions. Typically, the antigens are foreign substances like proteins and polysaccharides, carbohydrates, exposed in the surface of the invading microbes. The antigen stimulates the production of its own antibody and specify antibody binding, which follows leads eventually and the destruction of the microbes. Rather surprisingly, single lymphocyte produce just one kind of antibody. Thus, to protect the body against all the possible kinds of diseases, there must be thousands of different lymphocytes, each capable of recognizing and responding to a unique antigen precisely as found over there. There are blood groups and transplantation. We'll talk about them as well. But right now, what are we going to talk about is to continue with the antibodies. When B cells are stimulated, they enlarge and reproduce to form clones in the same ways as T cells, but the clone cells differentiate further into plasma cells, which remain within the lymphoid tissues. They have a well-developed rough ER and are specialized to produce antibodies, which are secreted at a rate of production maintained for several days, and after which the cell dies. The antibodies pass into the bloodstream and can have a number of different effects, ranging from the destruction of lysis and microbe, and then the opsonization, in which the antibodies attached with the surface of microorganisms make it more susceptible. Then there are memory cells from each clone remain in the lymphoid tissue, so that the second exposure of the same antigen causes much more rapid and vigorous in the response. Occasionally, the response of the repeated exposure is so dramatic that when we talk about the clones, we certainly talk about them all the way. The presence of circulating antibodies and of the memory cells in the lymph nodes gives lasting protection and immunity against disease organism. The same immunity is now commonly produced by the vaccination as well, in which dead or weakened microbes or purified antigens are isolated from them and they are injected into the body. This simulates the appropriate hemophagocytes just as to effectively contradict and battle the disease.